Today I'm going to talk about my favorite metabolic pathway, the pentose phosphate pathway. It's important because it makes molecules that are the starting point for a bunch of different biosynthetic pathways. It's got all kinds of interesting regulation, mostly driven by Le Chatelier's principle, which is actually why I think it's my favorite. So let's dig into it. We're going to be talking about this in a couple of videos. The first one, we're going to focus on what's called the non-oxidative part of this. And then we're going to go on and talk about the oxidative part and then how these all work together in subsequent videos. So for the moment, non-oxidative pentose phosphate pathway. So let's situate us inside of metabolism as a sort of network. I've been talking about this glucose transit network, everything over here that's kind of interconvertible with glucose. We've talked about things like glycolysis and gluconeogenesis, which move us up and down this arm, and glycogen storage, which allows us to store glucose for later. Today, we're finally getting over here to this complicated what's happening over in this network. That doesn't look the same as the other metabolic pathways. In terms of what is available to glucose in a cell, what it's used for, the non-oxidative PPP or pentose phosphate pathway is important for generating anabolic starting materials for things like nucleic acids and amino acids. These are often derived from parts of the pentose phosphate pathway. And here it is, well, at least a summary of bits of it. So let's do a quick anatomy lesson, and then we'll dig into some more of the details. So first of all, you might look over here and think this looks kind of familiar. And the reason it's kind of familiar is this is I was going to write gluconeogenesis. It is that, but it's glycolysis typically as written, right? So these are enzymes for taking a glucose 6-phosphate, fructose 6-phosphate, right? And then we can run it backwards. We can also go this direction, which would be gluconeogenesis. The reason that we're putting this in this description of the pentose phosphate pathway is you'll notice the pentose phosphate pathway actually has G3P and fructose 6-phosphate as part of it. So these are participating in both glycolysis and in the pentose phosphate network. This network here, which I'm showing in blue, is the non-oxidative part. And that's going to be the focus of today's video. And then this thing up here is what's known as the oxidative part. And we're going to follow that up with a second video later. So it's this intimate connection between this pentose phosphate network thing and glycolysis in this other non-oxidative pathway. I also want to point out that R5P is the starting point for all nucleic acid synthesis. So basically, if you're duplicating your genome, for example, and making a lot of nucleic acids, this R5P is the molecule that you're going to have to make a lot of. And then other metabolites go on to do other things. I'm just highlighting one of them. E4P is important in amino acid synthesis. So again, this network is used to generate metabolites that go on to be used in biosynthetic pathways. Just to summarize what I said there with some written form, non-oxidative PPP generates useful anabolic metabolites, so anabolic building new things, and metabolites, there we go. And the other thing you might have noticed is that the non-oxidative PPP is a network rather than a linear pathway. So unlike, say, glycolysis, where we can say A goes to B, goes to C, goes to D, this is this network of metabolites that get passed around. So what are those metabolites? And let's show these here. There's a lot of them, so I'm going to hide. These are the molecules that I've shown in written form. So we've got G3P, E4P, Ru5P, X5P, R5P, F6P, and S7P. Lots of things. You might be saying, well, how do I, what are some patterns? One of the things you might observe is they all have a P at the end. And that's because every single one of these has a phosphate stuck onto its highest numbered member. So if you think about these are all sugars. So they have on this side an aldose, in this case, aldose, or a ketose, aldose, ketose. And then that by sort of definition in sugar nomenclature is the one sugar. That means the phosphate is on the opposite side of the sugar from that. So it's at the highest numbered end. So if we see G3P, that means we have a phosphate at the 3 position of this sugar. Conveniently, that also then tells us the number of carbons that are in each of these, right? So G3P has 3 carbons, E4P has 4 carbons, R5P has 5 carbons, S7P has 7 carbons, for example. So what are these metabolites? They are sugars that have a single phosphate stuck onto the end of them that have a variable number of carbons. They also vary in whether they're aldose or ketose, whether the carbonyl is here or here. Okay, cool. What else can we talk about with this? Well, what these PPP metabolites do is they link different metabolic pathways. So we can think about this as sort of a switching station, a central exchange inside of metabolism. So G3P and F6P, these link into glycolysis. What do we see here? Ru5P links into the oxidative PPP. So it's a direct connection to that chunk of the pathway. R5P links into nucleic acids, biosynthesis. And E4P links into amino acid, biosynthesis. So again, even though there's a lot of metabolites and it feels like this complicated mess, they really are important for the understanding biochemistry. All right, what are the enzymes in the PPP? So it turns out non-oxidative PPP is built around 4 different enzymes. Transketolase, transaldolase, Ru5P isomerase, and Ru5P epimerase. So there's just 4 enzymes. And all of these reactions are fast and reversible. So when these enzymes are present, they're going to equilibrate effectively. So the left and right side of each reaction equilibrates basically so fast that when we look in a cell that's at steady state, that reaction will effectively be at equilibrium. All right, what do these enzymes do? So the transketolase transfers, complicated word there, these hydroxyacetyl groups. And so what we can see here is it's taking F6P, which is a ketose, and G3P, which is an aldose. And all that it's doing is it's taking off this hydroxyacetyl group, sticking it on to the aldose sugar, and what that generates is now 2 new sugars, a E4P and an X5P, right? So E4P and X5P are just the result of reshuffling the carbons that are present. Another thing that's important about this, if you look, is you can just count the number of carbons and see how the reaction works, right? So see what's going on here. So we've got 6 carbons here and 3 carbons here. We're going to take a 2-carbon chunk off of F6P and we're going to stick it onto G3P. So now we go to a 4-carbon and a 5-carbon. So this is always a 9 carbons, right? 3 and 6, and 9 carbons, 4 and 5. And so the last thing I'll note about this, and then I'll stop belaboring this, is it goes from ketose aldose to aldose ketose. So it's always going to be an aldose sugar and a ketose sugar. We're basically swapping them one for each other. Okay, so that's the transketolase. And it actually catalyzes, this one enzyme actually catalyzes 2 different reactions inside the PPP. Both of them are these transfer reactions, but one is a 6,3 going to a 4,5, and the other is a 5,5 going to a 3,7. Okay, so then again, it's just taking this 2-carbon chunk off and moving it around. But it can do it on substrates that are 6 and 3, or 5 and 5, and then they're paired partners, so 4 and 5 and 3 and 7. Okay. The second enzyme I mentioned is this one called transaldolase, and it transfers a dihydroxyethyl group. And so what is a dihydroxyethyl group? It is a 3-carbon version of basically what we saw with the transketolase. And so it's going to take things like S7P, which has 3 carbons there, and G3P. And then it's going to swap that with each other, generating E4P and F6P. And hopefully what you're seeing here is you're like, wait, these enzymes have similar substrates to each other. We've seen all these substrates before, yet we have different enzymes that are doing different moves on them. Just as a fun aside, both of these enzymes use what's called a ping pong mechanism, which is a really common mechanism in biochemistry, which I'll just highlight at a very high level. Basically, what it is the first substrate comes in. Wow, English. What happens is the group that is being transferred ends up being covalently attached to the enzyme, and the first substrate then falls out. Then the next substrate comes in, and the covalently attached transferred group gets put on. So it's ping pong because there's binding with the first substrate, chemistry, that leaves. Binding, then the second substrate binds and leaves. So ping pong, ping pong. So really common mechanism across biochemical enzymes. Really good example here with these transketolases and transaldolases. Okay, last two enzymes, and then we'll talk about how this sort of plugs together. That is Ru5P isomerase and Ru5P epimerase. Basically, these isomerize pentose phosphate sugars. And in particular, they take these 5-carbon sugars, ribulose 5-phosphate and ribose 5-phosphate and xylose 5-phosphate, and they just change around various things. So the epimerase, various things, that was very vague, sorry. The isomerase takes the ketose and turns it into an aldose. That's just like we've actually seen it, a similar reaction before when we saw the change for fructose to glucose, right? Glucose and fructose. All we're doing is we're changing where the carbonyl is. And then the epimerase is flipping a single stereocenter in Ru5P and X5P. So that's the only difference between these two molecules is that one stereocenter. Okay, so what we've seen is we have a bunch of enzymes that are fast and reversible and catalyze the exchange of a bunch of different reactions, right? They either move they move 2 carbon units and 3 carbon units, and they isomerize things, and you put them all into a mix, and what happens is you end up building a responsive network. And so this responsive network here is going to be able to change what concentrations of things are around just based on Le Chatelier's principle, right? If I build up a ton of R5P, let's say, so this starts going up, well, it's going to start exchanging, right? This reaction right here is going to have an excess of reactants, so we're going to start driving the reaction this way. We're going to start generating S7P and G3P. And if that means G3P's concentration starts going up, so maybe this reaction goes this way and we start speeding up glycolysis. So as different pathways make or break down members of this pentose phosphate network, the whole system can respond in a coordinated sort of fashion. And what this basically means is, depending on cellular needs, the pentose phosphate pathways can process different combinations of anabolic intermediates, and then, as we'll see with the oxidative part, reducing power, allowing it to access all kinds of interesting configurations. The last thing I want to point out is that cancer cells are actually big fans of the pentose phosphate pathway. I should do a quick move here, sorry. They're big fans of the pentose phosphate pathway because they need a lot of metabolites to grow. And so as they're, say, dividing, they are going to need a ton of ribose to make nucleotides, which means their pentose phosphate pathway tends to be operating at a higher level than many other tissues nearby. And that means that metabolically speaking, if you want to differentiate cancer cells from non-cancer cells, a really good way to do it is to look at the state of their pentose phosphate network. Okay, so that is all I have on that. I just have to do a quick move here. There we go. That's all I have on that. In the next couple of videos, we're going to look at the oxidative pentose phosphate pathway and then how these integrate into interesting metabolic modes. Hopefully that was cool and interesting as it is, and I will see you later.